The benefits of Amla. This is printed in the Pharma Innovation Journal. Pharmacological properties and health benefits of Emblica officinalis. Amla is a medicinal and nutritional tonic that contains essential amino acids and vitamins. When compared to other citrus fruits, it's a particularly good source of vitamin C and minerals. And although all parts of Amla are used medicinally, the fruits are more extensively utilized for the treatment of numerous infectious and non-infectious disorders, either alone or in combination with other traditional herbs. E. officinalis is one of the most researched plants, and findings indicate that it includes tannins, phenolic compounds, and alkaloids. The fruits of E. officinalis have been shown to have greater levels of vitamin C as well as higher amounts of most minerals, protein, and amino acids, such as glutamic acid, proline, aspartic acid, alanine, cysteine, and lysine. The hydrosoluble tannins, Emblica A and B, penglogocan and pendiglocan, are more abundant in the fresh pericarp of E. officinalis. Reports of the pharmacological research on Amla reveals its antitussive, analgesic, anti-atherogenic, adaptogenic, gastro, cardio, nephro, anti-cancer properties, and neuroprotective. Amla is also reported to possess immunomodulatory, free radical scavenging, chemopreventative, anti-inflammatory, anti-mutagenic, and antioxidant properties. These properties are effective in the treatment and prevention of numerous diseases like atherosclerosis, cancer, peptic ulcer, diabetes, anemia, heart disease, liver disorders, and various others. The antioxidant properties of Emblica. Free radicals are the building blocks of any biochemical reaction, which are continually created by the body's routine utilization of oxygen. Naturally, there's a dynamic balance between the number of free radicals produced in the body and the amount of antioxidants available to quench, scavenge, and shield the organism from their harmful effects. In India, a traditional medication, including Emblica officinalis, was developed and utilized as a health tonic and has exhibited strong antioxidant activity. Emblica officinalis contains antioxidants that are heat stable and water soluble and only partly extractable with ether. Through oxidative processes are necessary for survival, they can harm the body's basic cells. Amla promotes cell viability by lowering free radical generation and increasing antioxidant levels. To combat oxidative damage, nature has provided sophisticated antioxidant defense systems, such as vitamin A, C, and E, as well as numerous enzymes such as superoxide dismutase, glutathione, catalase, peroxidase, and others. In a research that incubated E. officinalis for 24 hours, significant reduction in liquid hydroperoxide and reactive oxygen species were reported. E. officinalis also boosted glutathione peroxidase levels, antioxidant enzyme activities, and antioxidant capacity. Furthermore, when given once daily for 7 days, the active tannoids of E. officinalis increased the activity of catalase, superoxidase dismutase, and GSH peroxidase. in the frontal and striatal cortex, as well as a reduction in lipid peroxidation in these brain regions. The complete fruit of E. officinalis is recognized for its antioxidant properties. However, the pulp and seed each have their own unique phytopharmacological functions. The pulp of the E. officinalis has more antioxidant capacity than the seed, with the values showing the lowest drug concentration in the respectively. The fruits of the amla, which constitutes Emblica A and B, are reported to have potent antioxidant effects. Amla increases cell viability by decreasing free radical production and raising antioxidant levels. The findings also suggest that the antioxidant activity of E. officinalis may be found in the tannoids found in the plant's fruits, which have vitamin C-like qualities but are not vitamin C. Another investigation claims that phytochemicals included in amla can act as an excellent metal ion chelator, preventing oxidative cascades, and in a rat model, the antioxidant activity of tannoid active components of amla was evaluated. Anti-diabetic properties of amla. Diet has a crucial role in the treatment of diabetes and its consequences. Numerous classical herbs have been documented to have anti-hypoglycemic and hypoglycemic actions in varied degrees. These actions appear to be achieved by higher insulin production via pancreatic cell stimulation, interfering with dietary glucose absorption, or insulin sensitization. On normal and naloxone diabetic rats, oral treatment of the extracts dramatically lowered blood sugar levels in 4 hours. In rats, E. officinalis and a concentrated portion of its tannoids are efficient at delaying the onset of diabetic cataract. Aldose reductase plays a role in the development of diabetes secondary consequences, such as cataracts. E. officinalis has been shown to be an effective AR inhibitor. Diabetic neuropathy is one of the most frequent microvascular consequences of diabetes, affecting more than half of all diabetic patients. In a study, the effects of a high flavonoid extract of E. officinalis in male Sprague Dawley rats with diabetic neuropathy from due to type 2 diabetes. In diabetic rats, the administration of amla extract increased tail flick latency in a hot immersion test and pain threshold level in a hot plate test when compared to the control group of rats. The anti-diabetic activity of E. officinalis was tested in diabetic rats. When compared to the metformin-treated group, the plant-extracted group showed a substantial drop in blood glucose. Furthermore, extracted doses of 200 to 400 milligrams resulted in a significant reduction in blood glucose and lipid levels, similar to the metformin-treated group. In uremic diabetic patients, the oral treatment of a 1-1 combination of epigallocalate and amla extract for 3 months increased the antioxidant defense as well as the diabetic and atherogenic indices. Cardioprotective properties of amla. Low-density lipoprotein cholesterol is a primary contributor to the development of atherosclerosis. Antioxidants are regarded as an excellent therapeutic treatment for atherogenesis due to oxidative stress E. officinalis has been shown in several preclinical investigations with laboratory animals to have cardioprotective and anticoagulant properties, suggesting that it might be a beneficial medication for delaying and/or preventing different cardio-related diseases. The presence of tannoids in amla, notably ellagic acid, Emblica A and B, gallic acid, and coralline, is thought to be responsible for this protective effect. Oxidized LDL is the primary causative factor in arthrogenesis. Therefore, antioxidants are often used to treat atherosclerosis. The findings imply that E. officinalis can slow the progression of atherosclerosis by reducing oxidative stress or suppressing the smooth muscle cell proliferation, both of which could be attractive therapeutic targets for atherosclerosis. Supplementing with extracts reduce the oxidative damage caused by a high-fat diet. According to the findings, E. officinalis extract has a cardioprotective effect in rats fed in a high-fat diet. In rats, the effects of prolonged oral administration of fresh amla fruit homogenate on a myocardial antioxidant system. An oxidative stress generated by ischemic reperfusion damage were studied. Chronic E. officinalis treatment causes myocardial adaptation and protects rat hearts from oxidative stress caused by RAJ by increasing endogenous antioxidants. The ethanolic extract of E. officinalis was studied for its cardiovascular preventative and lipid-lowering qualities. The lipid profile of rats fed a high-fat diet was shown to be changed, and it was found to be reversible when the extract was supplied. After supplementing with E. officinalis, a group of high dietary fat-fed rats showed improvement in cardiac autonomic malfunctions, and electrophysiological assessment indicated changed heart rate and sympathovagal balance. Anti-cancer properties of Emblica. Natural medicines derived from plants now make up a significant share of commercial anti-cancer treatments. Medicinal plants or active compounds of medicinal plants have a function in cancer care by altering the activation of numerous genes. One of the most frequent malignancies in women is breast cancer. Breast cancer risk has been linked to lipid metabolizing enzymes, lipids, and lipoproteins. Kapla-Mutha K has a city formulation that includes EO, sempercarpus, and honey. On treatment with the Ka and Sa, the raised levels of free cholesterol, total cholesterol, triglycerides, free fatty acids, and phospholipids, as well as the lowered levels of ester cholesterol detected in cancer-stricken animals, were reduced to near normal levels in kidney, plasma, and liver. The effect of amla fruit extract on tumor incidence was tested in a process involving 2 stages of skin carcinogenesis in Swiss albino mice, which was triggered by a single administration of 7,12-benzene atrazine, and then enhanced by recurrent application of croton oil until the experiment's completion in 16 weeks. Tumor incidence, tumor burden, tumor production, and a cumulative number of papillomas were all greater in the control mice than in the experimental animals. Another study discovered that the anti-metastatic efficacy of amla extract is essential in lowering cancer migration, cell invasion, proliferation, and adhesion in a time of dose-dependent way. Both MMPT and MMPN expression decreased in this example, indicating a biological mechanism for autometastases. In addition, many studies have shown that E. officinalis may protect against chemo and radiotherapy side effects. Antimicrobial properties of Emblica. Infectious illnesses are a leading cause of morbidity and death throughout the world. Despite significant advances in microbiology and the management of germs, periodic outbreaks of epidemics caused by drug-resistant bacteria and newly identified disease-causing microbes contribute a significant threat to public health. These alarming health trends necessitate a global effort to discover creative ways for the prevention and treatment of infectious illness. Chemical compounds derived from medicinal plants have been used as models for several therapeutically established medications for more than a century and are currently being re-examined as antimicrobial agents. Various research implies that E. officinalis have substantial antibacterial action, which might be a valuable lead for the creation of cost-effective and safe treatments. Antimicrobial activity was tested in aqueous and sequential extracts of E. officinalis fruit pulp and Ocimum sanctum leaves and stems extracts. The results were linked to the conventional antibiotics amoxicillin and amphotericin B. The methanolic extract of E. officinalis was shown to be more effective than the leaf and stem extracts in preventing all pathogenic microorganisms. E. officinalis extracts, both alcoholic and aqueous, were shown to be effective against a variety of human diseases, including bacteria, viruses, and fungus. Activity against gram-positive bacteria appears to be greater with just minimal efficacy against fungi. Induced Klebsiella pneumonia in mice reacted to powdered fruit nutrition in the long term. In the near term, however, colonization was not avoided. The phenolic contact has been linked to action against herpes simplex viruses 1 and 2, while the phyloembellicin B isolated from the plant's roots has been linked to activity against the Coxsackie virus. And from the methylonic extract of E. officinalis, alkaloids were extracted, which showed strong antimicrobial activity against gram-positive bacteria, such as Staphylococcus aureus, Bacillus cereus, Circinus luteum, Bacillus subtilis, as well as gram-negative pathogenic bacteria such as Salmonella typhi, E. coli, Salmonella, Shingilla bardi, Pseudomonas aeruginosa, and some strains of fungi, Saccharomyces cerevisiae, Candida, and Aspergillus niger. Anti-inflammatory properties of Emblica. Many plant-derived substances have been demonstrated to have significant anti-inflammatory properties. As a result, they represent potential compounds for the creation of novel medications, especially for the treatment and/or management of chronic inflammatory conditions, such as asthma, rheumatism, arteriosclerosis, inflammatory bowel illnesses, and so on. The application of intraperitoneal of E. officinalis hydroalcoholic extract of various dosages is reduced granuloma formation and significantly prevented rat paw edema against all phylogistic agents. Though the extract caused entire-out anti-inflammatory responses in research models at frequent doses and demonstrated equivalent benefits to typical anti-inflammatory medicines. The aqueous fraction of Amla methanolic extract identify activity against carrageenan as well as dextran-induced edema in rats, and then the same fraction was tested on the formation of inflammatory mediators, specifically platelet activating factor, thromboxane, leukotriene, N-formamide, and LTB4 and N-formamide. Human PMNs were inhibited by an aqueous fraction of the Amla, but inflammatory mediator production was unaffected during blood clotting, suggesting that anti-inflammatory effect does not entail suppression of lipid mediator synthesis. In vitro investigations utilizing bronchial epithelial cells taken from a cystic fibrosis patient, on the other hand, showed that pro-inflammatory cytokine expression was inhibited. Conclusion. Amla is a well-known Indian medicinal herb which provides numerous health benefits. Amla fruit is widely distributed in tropical and subtropical countries. All parts of this fruit possess medicinal properties, particularly the fruit, which is used in Ayurveda as a powerful rasayana, and in medicine in the treatment of diarrhea, jaundice, inflammation, and several other ailments. The antioxidant and phytonutrients help in fighting against free radicals. Amla possesses anti-cancer properties because of high concentration of polyphenols. The Amla fruit contains carbohydrate, pectin, protein, and fiber with high moisture content. Amla is a rich source of vitamin C. The juice extracted from the Amla fruit is observed to contain high amount of vitamin C. The main components of Amla fruit are gallic acid, ellagic acid, gibberellic acid, gibberellic acid, Emblica acid N, A, Emblica B, phenylgluconine, pendugluconine, elogactinin, trigallyl glucose, coralegan, and isotretinoin, and many more. It's also a rich source of organic acids like citric acid, fumaric acid, malic acid, and tartaric acid. Amla is a rich source of various minerals like aluminum, calcium, zinc, iron, chromium, copper, lead, lithium, magnesium, manganese, molybdenum, nickel, phosphorus, potassium, and sodium. It helps to control digestive problems, strengthens cardiovascular activities, and makes our immune system strong, improving the eyesight, making our hair shine and strong. And by massaging Amla oil to the head, we can get better and proper sleep. It also will regulate the growth of hair follicles and help to prevent from premature graying of hair. By soaking Amla in water and washing the eyes with that water, it improves the eyesight, and drinking that water early in the morning will remove constipation. Amla is available in the market in different forms like Amla juice, Amla candy, Amla jam, Amla bar, Amla chutney, and pickles, Amla sauce, etc.